It seems like everyone is worried about cholesterol, and this might be for a good reason. You hear it from your doctor, and it's tested through that fasting lipid panel you have to do. It's already bad enough that this is a blood draw, but for most people, there's almost always something wrong with this test grouping. In this video, I'll explain the different parts of a lipid panel and what you need to know about each of them. Let's get right into it. You might be wondering what cholesterol even is, and it's a fat that has some pretty important jobs in your body. One of these functions is to maintain the fluidity of the cell membrane. It also serves as sort of a template for things such as steroid hormones, bile acids, and vitamin D. Look at how similar their structures are. With these interesting functions, you may be even more confused as to how cholesterol would be a bad thing. It's actually regulated by the liver, which makes enough cholesterol for bodily functions. However, we can also get cholesterol from food. For example, if the only cholesterol you eat is from an egg yolk, which happens to be around 200 milligrams of cholesterol, your body should produce an extra 800 milligrams of cholesterol. Some scientific evidence shows that dietary cholesterol alone in the presence of a healthy diet will not negatively affect blood lipids. My tip of the day is to eliminate simple sugars from the diet to improve cholesterol, since these may have a greater effect on your cholesterol levels. The body has systems to prevent excess cholesterol, but sometimes these can become overworked or just don't work properly. This excess cholesterol in your blood is problematic since it will settle in your arteries as plaque, which can lead to some life-threatening problems down the line, such as a heart attack or a stroke. If we look at the lipid panel recommendations, we see that normal is less than 200 milligrams per deciliter, borderline high is around 200 to 239, and then high is anything above 240. Sadly, cholesterol usually increases as people get older, and especially in postmenopausal women. Since there are many processes that go into cholesterol regulation in the liver, if there's a problem in one of these pathways, this can cause increased cholesterol levels. Some of these mutations can be inherited, such as familial hypercholesterolemia, which is a mouthful, and these will cause problems in clearing LDLs, which I'll talk about next. I'll start with the average recommendations for LDL levels. Optimal levels are usually around less than 100 milligrams per deciliter. This is the goal for people with diabetes or heart disease. Close to optimal is around 100 to 129. Borderline high is 130 to 159. High is 160 to 189. And very high is 190 or higher. LDLs, or low-density lipoproteins, are known as the dreaded bad cholesterol. But like many negative things, they're only a problem if you have too much. In fact, LDLs are important to your body and move cholesterol around the body to where it is needed, but become bad when you have too many of them circulating in your blood. They can combine with other substances and build up on the walls of your arteries. This builds up and gets bigger over time, and this plaque growth is called atherosclerosis. There are a few reasons that your LDLs may be high. One risk factor is weight and lack of physical activity. Being overweight puts you at a higher risk for increased LDL levels. The other reasons are the same I mentioned for cholesterol, such as age and genetics. There are currently a few medications that are used to decrease LDL levels. These are primarily HMG-CoA reductase inhibitors. Common ones include statin variants. HMG-CoA reductase is one of the enzymes in the production of cholesterol. So these medications basically slow down cholesterol production in the body. Now that you know what the bad cholesterol is, let's talk a little bit about good cholesterol, or HDLs. This type of cholesterol is actually good for you because it lowers your risk of heart disease. The higher the number, the lower your risk. 60 milligrams per deciliter or above is considered the level to protect you against heart disease. HDL means high-density lipoproteins, which means they are made up of mostly protein. It is good because it takes extra cholesterol out of your bloodstream and sends it back to the liver, which breaks it down further. For the most part, if you want this number higher, it's recommended for men to have an HDL level of at least 40 milligrams per deciliter and women to have at least over 50, which may be one of the reasons why women tend to live longer than men. There are a few things we know that will decrease your HDL levels, so it's best to avoid these if possible. Smoking will cause your HDL levels to be lower and in turn cause LDL and triglyceride levels to rise. It's been said countless times, but don't smoke. There's far too many negatives. Anabolic steroids have also been known to lower HDL levels, so you may want to reconsider if those extra gains are worth it or not. Another factor that lowers HDL levels is metabolic syndrome. This is a grouping of conditions that include obesity, high blood pressure, and high blood sugar. The best way to raise your good cholesterol is physical exercise, and it's been found that it can be increased with around 30 minutes of exercise daily. Other ways to increase it include improving your diet and medications that cause increased HDL levels or decreased LDL levels. However, it's important to note that the current research shows that HDL increase through medication does not necessarily give you the same benefits. Higher does not always mean better. The HDL still has to be efficient. One very interesting circumstance is that people with extremely high HDL levels, over 100, tend to be at a higher risk for heart disease. This is usually a genetics issue but can be caused by biliary, cirrhosis, and thyroid disorders. However, for the most part, people who have naturally high levels of HDL cholesterol are at a lower risk of heart attack and stroke. On to triglycerides. High levels of triglycerides are linked with a higher heart disease risk. Here are the adult ranges. Normal falls less than 150 milligrams per deciliter. Borderline high is around 150 to 199. High is 200 to 499. And very high is anything above 500. Since cholesterol cannot travel through your blood by itself, it needs to be attached to other molecules. I already told you how it joins up with proteins, but it also teams up with the triglycerides to form the lipoproteins. Let's get a little bit more into triglycerides. They mostly store energy, but also provide insulation to cells and help absorption of fat-soluble vitamins. Like LDLs, too many triglycerides is a bad thing. High levels of triglycerides in the blood is called hypertriglyceridemia. This can be genetic, but it can also be due to eating too much. When you eat, your body uses whatever it can and then stores the rest. Since triglycerides are so great at storing energy, a lot of excess calories will be stored as such. When you need energy later, hormones will release these triglycerides. This is usually due to a diet that contains too many carbohydrates, so make sure you try to avoid those simple carbohydrates. Like LDLs, high triglycerides may contribute to the hardening of your arteries and the walls of your arteries, which increases your risk of stroke, heart attack, and heart diseases. It is important to note that insanely high triglycerides can also cause pancreatitis. I hope you learned something about cholesterol or lipid panels in this video. If you did, be sure to leave a like and subscribe. Thanks for watching.